The image of God. What is the image of God? Whatever the image of God is, it's subject to much debate. And the purpose of this video is not to delineate exactly what the image of God is, but to point out how important it is and that it doesn't need to be messed with. Whatever the image of God is, it's eternal, for we're created as eternal beings. Again, we will live for eternity, eternal life or eternal damnation. Either way, we're eternal. And I believe part of this eternity is rooted in our DNA. The Lord is eternal. Who can figure all that out? The DNA is very complex.
So it's an accurate model of DNA. If we unwind the double helix and unzip the two strands, you see these things that look like teeth. Those are the letters of genetic code, the 25,000 genes you've got written in your DNA. This is what they typically talk about, the genetic code. This is what they're talking about. But I want to talk about a different aspect of DNA science, and that is the physical nature of DNA. And it's these two strands that run in opposite directions for reasons I can't go into right now, but they physically run in opposite directions, which creates a number of complications for your living cells, as you're about to see, most particularly when DNA is being copied. And so what I'm about to show you is an accurate representation of the actual DNA replication machine that's occurring right now inside your body, at least 2002 biology. So DNA is entering the production line from the left-hand side, and it hits this collection, this miniature biochemical machines that are pulling apart the DNA strand and making an exact copy. So DNA comes in, it hits this blue donut-shaped structure, and it's ripped apart into its two strands. One strand can be copied directly, and it can be seen spooling off down to the bottom there. But things aren't so simple for the other strand because it must be copied backwards. So it's thrown out repeatedly in these loops and copied one section at a time, creating two new DNA molecules. Now, you have billions of this machine right now whirring the right way inside you, copying your DNA with exquisite fidelity. It's an accurate representation, and it's pretty much at the correct speed for what it's occurring inside you. But I've left out error correction and a bunch of other things. This was work from a number of years ago. Thank you. This is work from a number of years ago, but what I want to show you next is updated science. It's updated technology. So again, we begin with DNA, and it's jiggling and wiggling there because of the surrounding soup of molecules, which I've stripped away so you can see something. DNA is about 2 nanometers across, which is really quite tiny. But in each one of your cells, each strand of DNA is about 30 to 40 million nanometers long. So to keep the DNA organized, to regulate access to the genetic code, it's wrapped around these purple proteins. I've labeled them purple here. It's packaged up and bundled up. All of its field of view is a single strand of DNA. This huge package of DNA is called a chromosome, and we'll come back to chromosomes in a minute. We're pulling out, we're zooming out through a nuclear pore, which is sort of the gateway to this compartment that holds all the DNA called the nucleus. All of this field of view is about a semester's worth of biology, and I've got 7 minutes, so we're not going to be able to do that today. No, I'm being told no. This is the way a living cell looks down a light microscope, and it's been filmed under time-lapse, which is why you can see it moving. The nuclear envelope breaks down. These sausage-shaped things are the chromosomes, and we'll focus on them. They go through this very striking motion that is focused on these little red spots. When the cell feels it's ready to go, it rips apart the chromosome. One set of DNA goes to one side, the other side gets the other set of DNA, identical copies of DNA, and then the cell splits down the middle. And again, you have billions of cells undergoing this process right now inside of you. Now we're going to rewind and just focus on the chromosomes and look at its structure and describe it. So again, here we are at that equator moment. The chromosomes line up, and if we isolate just one chromosome, we're going to pull it out and have a look at its structure. So this is one of the biggest molecular structures that you have, at least as far as we've discovered so far, inside of us. So this is a single chromosome, and you have 2 strands of DNA in each chromosome. One is bundled up into one sausage, the other strand is bundled up into the other sausage. These things that look like whiskers that are sticking out from either side are the dynamic scaffolding of the cell. They're called microtubules, but name's not so important. But what we're going to focus on is this red region. I've labeled it red here, and it's the interface between the dynamic scaffolding and the chromosomes. It is obviously central to the movement of the chromosomes, but we have no idea really, as to how it's achieving that movement. We've been studying this thing called the kinetochore for over 100 years with intense study, and we're still just beginning to discover what it's all about. It is made up of about 200 different types of proteins, thousands of proteins in total. It is a signal broadcasting system. It broadcasts through chemical signals, telling the rest of the cell when it's ready, when it feels that everything is aligned and ready to go for the separation of the chromosomes. It is able to couple onto the growing and shrinking microtubules. It's transiently, it's involved with the growing of the microtubules, and it's able to transiently couple onto them. It's also a tension sensing system. It's able to feel when the cell is ready, when the chromosome is correctly positioned. It's turning green here because it feels that everything is just right. And you'll see that there's one little last bit that's still remaining red. And it's walked away down the microtubules. That is the signal broadcasting system sending out the stop signal, and it's walked away. I mean, it's that mechanical. It's molecular clockwork. This is how you work at the molecular scale. So with a little bit of molecular eye candy, we've got kinesins, which are the orange ones. They're little molecular courier molecules walking one way. And here are the dynein. They're carrying that broadcasting system, and they've got their long legs so they can step around obstacles and so on. So again, this is all derived accurately from the science. The problem is we can't show it to you any other way.
DNA changes according to its location. Depending on the location of the DNA with other DNA is how our bodies respond. So it's far more complex than man just quoting, hey, we've mapped the entire genome. It's more complex than that. It involves the location of the DNA. The location of the DNA also changes over time, which adds another dimension.
There are these master computer programs that have a tremendous downstream effect. They control the development of the embryo in these amazing ways.
So it's like we have these programs that are creating all this diversity, but there's a master program, almost like an operating system.
Absolutely. That operating system, actually, scientists have compared the operating system of a simple bacterium to the Linux operating system.
Which is very, very complex.
Very complex, but when we look at it, there's a lot of computer programming in life. And so what we've seen is just like in our operating systems, life has some master modules and then has some middle level programs that control all the output. We call that proteins and sugars and action. Computer systems, they've got your operating system that controls a lot of middle level managers, do this to the sound card, do this to video card, that controls all the outputs. But it's really funny, when we look at the way life is structured, it's a lot more optimally designed. You have less high-level programs controlling less middle-level programs controlling a lot more outputs. Our computer programs are top-heavy. We have a lot of stuff up here controlling a little bit of stuff down here because we're not quite as smart as God. If you want to get that starfish to turn into a radically different organism, you don't tweak these little things. You have to tweak the high-level operating system, and that usually causes a downstream catastrophe. I mean, imagine if you're programming a computer, right? You can change a 1 or a 0 in an output, and maybe now your letters are blue instead of white on the screen. Big deal. But if you want to randomly change the core operating system, how many changes are possible that don't cause disaster? I mean, almost all changes would ruin the whole system.
From a computer scientist perspective, I can tell you, we call that system software sometimes. That's the fundamental heart of that whole system. If you have a problem down there, the whole thing breaks down.
And when someone wants to change that, how many people have to think for how long to make a simple change to the basic programming?
That's tough, because once you start monkeying with that operating system, with that system software, you have to be very careful because you'll upset the whole thing.
Yeah, it's dynamic programming. You have a gene, and this piece is used over here or over there or over there. And there's little teeny programs inside the DNA that control when and where and how to use that piece. But just recently I read a paper about actually shifting of the information in the genes. So if you start at letter number 1, you can read out this information. If you start at letter number 2, you get a totally different information. How on earth did that evolve like that? I mean, if you think of, if you read a story, maybe you're reading a story talking about some swashbuckling pirate. But if you start the second letter, it's a chocolate chip cookie recipe. We can't write that. There's no way we could intelligently program multiple levels of information into the same story. And that's what we see in life. And if we can't do it intelligently, it's not going to happen randomly.
We're talking about something that is beyond imagination in terms of the complexity, even from the standpoint of the kind of things we've done with software, and we've done some amazing things with software. But that appears to be is not even close to what you're talking about.
I like to say the genome is four-dimensional. In software, we write in lines of code.
Correct.
Well, in mathematics, we learned a line is a one-dimensional object. It just has length. So you could actually take a computer program and just write from left to right for millions of characters. Now, on our screens, we put carriage returns in there so we can read it. But the computer doesn't know that. It ignores the carriage returns. It's just a line. Well, DNA is a line. So in the naive concept of DNA, we had a line that had information in it. But it's not simple like that. Because this piece of DNA makes a little protein that comes over here and sticks on this piece of DNA over here, which turns on or turns off a gene.
Oh my goodness, it's like self-modifying code.
Oh, it's worse than that. Because this piece of DNA over here makes just an RNA that goes over here and interferes with this gene's RNA. They stick together, they interfere, they conflict with one another, they turn things on, they turn things off. And if you want to draw that out, you would need a sheet of paper, a very big sheet of paper. You'd have to write all the letters of DNA out on all 3 billion of them. It would take, I calculated, about 850 Bibles as one human genome. And then you have to draw lines or arrows from one part to another part, because this part turns this part off, this part interferes with this part enhances this. It's this huge two-dimensional interaction network, and that's why you have a two-dimensional genome.
So it sounds like, I mean, let me stop you
All
for
right.
a second, because this is really amazing to think about this, because I think in terms of a computer program that it's fairly static. I mean, the instructions are there, but you're talking about a program that is reprogramming itself, that's modifying its own instructions.
Oh, wait till I get to the 4th dimension.
Oh, okay.
It's because of the 3rd dimension first. The genome also folds into a 3-dimensional shape. And so a 3D, the 3rd dimension, is actually the shape. And the genes that are buried inside this ball of DNA, they're not active, they're turned off. The genes that are exposed are the ones that are used. So whoever programmed that string knew when it folded up which genes will be available at what time.
I say, are you saying that when this instruction set folds onto itself, it creates a whole new set of instructions?
Yeah, absolutely. And
That's mind-boggling.
The information in that 1st dimension, that linear string, has to be organized in such a way that when it folds into the 3rd dimension, it still works.
Oh, that's amazing.
The 4th dimension is time.
And how does that work?
The genome changes shape over time. Remember I said that some genes are buried?
Yes.
And some are exposed? Well, you need those buried genes at some time. And so at different stages of development, or sleeping versus waking, or stress versus non-stress, or maybe you eat something that's bad for you, and your liver says, I can get rid of that toxin. Now, your earlobes, they don't care, they don't know what to do, but your liver says, I know what to do. The chromosomes in the liver will change shape, expose that new protein gene, make copies of it, build a brand new protein that can kill off that toxin, and when it's not needed anymore, they'll change shape again and fold back.
Oh my goodness.
Some of the information in the genome is like scaffolding in the building. The reason this piece of DNA is here is because when it falls into the 3 dimensions, it needs these 2 genes to be next to each other.
Right.
So this stretch here might not have a functional protein associated with it, but it still has a function. It's very important. So most of the so-called junk DNA has been brought into the functional category, just not in the way the standard paradigm predicted. But the interaction networks in that 2nd dimension, they change. Because this gene turns off, well, that affects this one over here. This one no longer talks to that one over there. But it's worse. It's even more complicated than that. It's the 1st dimension, that linear string.
Yeah.
The program changes. The
now we're
computer
getting-
software people, they don't like programs that dynamically rewrite themselves. You get all sorts of catastrophe, but we've learned that in the human brain cells have different genomes to other brain cells. There are these little pieces of DNA that they make a circle and they pop out and they go over and they float somewhere else in the genome and they stick themselves in there and they turn genes on and they turn genes off. And now we have different pieces of DNA in different brain cells, and that directs what type of brain cell it will become. But our liver cells, they have different genomes also. There's a lot of chromosomal duplications that happen in the liver. Because if you need a biochemical pathway and a lot of it, well, make extra copies of those protein genes. But different liver cells have different copies of different chromosomes.
Rob, that's so far beyond anything that we know, even in our most complex software systems, that it's almost beyond imagination to think that someone would look at that and say it all happened by chance.
Yes, and it only brings glory to God.
It does.
Because the more complex it becomes, the less possible it is to explain with natural, simple mutational processes. And we realize that God is so far above us in intelligence. We don't program computers like that because we're not that smart. But He made us in His image. Systems like that don't come about through a stepwise process. You've got things that have to exist or life cannot exist. And they're intrinsic and important and found throughout all of life. It has to be not built up stepwise, but the whole system laid out, spun up, and then God let go.
Yeah.
And there it works.
Wow.
Like Mickey Mouse, the Sorcerer's Apprentice, messing with the engines of the universe. Pandora's box, it all gets messed up. Who can fix that? Do you want to take that gamble in your body? There's something very unique going on now around the world. There are things that you can put inside your body through injections.
Influenza virus. It's unusual because it's an RNA virus, yet it has to get its genome in the nucleus to replicate. Why that's so, we'll talk about next time. It does so by being transported through the nuclear pore. Remember, the virus uncoats in the endosome, and then its RNPs, its ribonuclear proteins, or its helicocapsids are really small, and they can fit right through the nuclear pore, which, as you know, can transport material of a certain size into the nucleus. Most viruses are too big to cross the pore. The exception are the parvoviruses, shown in the last panel, which are small enough to fit right through the pore, and they can be transported in, and then they uncoat their DNA in the nucleus. But other viruses are too big, and so they have to take other strategies. Here is what herpes virus does. Remember, we talked about the portal at one of the five-fold axes of herpes virus last time. It's a hole through which the DNA comes. This virion is docked onto a nuclear pore, and the DNA is being threaded into the nucleus. What the signal for that is, we don't know. It would be a very interesting thing to find out. Adenovirus we talked about. Remember, adenovirus begins to be partially disassembled in the endosome. It breaks through the endosome, partially disassembled, that's shown here, moves to the nuclear pore, and then it docks on it, and again the genome is pulled in. There's some evidence adenovirus DNA is coated with various proteins, and there's some evidence that these serve as tethers for nuclear proteins to pull the DNA into the nucleus. So 4 different ways of getting nucleic acid into the nucleus.
The DNA is masterfully written. In fact, it may have a signature by the artist himself.
I wrote a book in 2003, it was released in 2004, called The God Code. What I did was I followed the instructions in a 3,000-year-old sacred text that had only been translated into English once. It's a Hebrew text called the Sefer Yetzirah, the Book of Creation. And it is as if someone were watching the moment that creation happened and recording it as a scribe, step by step. And it's language, it's words. I said, what would happen if I took those words and I converted the words into the periodic table of elements? So rather than taking fire and water and air and earth to make a human, what if we took, and there's a logic to this, what if we took hydrogen, nitrogen, oxygen, and carbon? And I began working with it from this perspective. What if that text was actually telling us a chemical formula? And if that's true, it means that the DNA in our bodies can literally be read like the letters of a page of a newspaper or a book. There is a message in the DNA of all life. So I came from that perspective. I did the work, I did the mathematics, and there is a message in the DNA of all life. It's in layers. The very first layer, the very first message in your body, every cell in your body has the same message. And the message, when it's translated, literally reads, the DNA literally reads, God eternal within the body. God eternal within the body. Now, why is that message in the human genome? I don't know. I just followed the instructions in the 3,000-year-old text to the T, and it's there. So someone figured out how to store data in DNA a long time ago. Now, immediately after I wrote that book, Japanese scientists came out with a report. They took bacteria and they began storing data in the DNA of bacteria, and they stored an entire library, a city block long, inside of the bacteria, and they let the bacteria reproduce 2,500 generations, and then they pulled the data out intact. It says that we are just now learning to store information inside of living DNA. If we're doing it now, it's possible that an advanced form of life has done that a long time ago. If we are the product of an intentional act, what better way to alert the beings that carry the message than to encode it within their very lives, within the DNA itself? Now that we have the technology, I get goosebumps just saying that. Now that we have the technology, we can literally read a message. And think about this. When we create something today, if you create a piece of art and you're proud of it, what's the last thing you do with that piece of art? Or a sculpture? Or you create a piece of electronics. Every tiny piece of electronics has an identifying mark on it somewhere. If we are the product of an intentional creation, I would not be surprised that within us is the mark of who or whatever is responsible in a way that would last as long as we do, that could not be destroyed like a temple wall, or could not be destroyed like an old manual or a text. It lives in the life itself. God eternal within the body. What does that mean? Who is God? I don't know what that means.
In fact, it may have a signature by the artist himself. This, again, is another area of controversy. It can be debated. And that's not the purpose of this video. You could debate it and come up with the right answer. Someone would disagree with you, and it's all very complex. Who can understand it? My point is, leave it alone. Changing the DNA, rewriting the DNA. If it's true that the Lord, as the author, the painter, has put his signature on our DNA, do you want someone else signing the DNA, changing the DNA? You shouldn't take that chance. And it's your responsibility to know what's going inside your body. The Lord Jesus wasn't too concerned about his disciples taking things into their body through the mouth. He speaks clearly what happens when you do that. And his disciples were accused of eating with unwashed hands. He wasn't concerned about that because he said it passes out through to the drought. When you get to the DNA, this is not just going through the eliminary system. This is going into your blood, into your cells. And it's your responsibility to know what it is and what effect it has. You can't pass that trust on to anyone else, regardless of what kind of initials they have after their name. It's your responsibility. Just say no.